Good morning, everyone. So we'll have a quick review of RNA splicing, and then we will go into some additional details of what happens to RNA after transcription. So we read about splicing, and we are reading about, we in the last class, we were reading about alternative splicing. That is about splicing that can occur in different ways. In that we saw uh, some of the things like this: if this is the normal one, and then there are other possibilities such as one exon might be skipped, or sometimes you may have an extended like this because of splicing happening at a different location compared to that of 5' splice site. In some cases, a complete intron could be retained, and in some other cases, you can have alternates. Either 1 and 2 will splice together, or 1 and 3 will splice together. And often these are called as isoforms because they have several stretches of identity, right? If this is the whole exon 1, for example, in these two, exon 1 is present in both the cases, and so they will have absolute identity in that part of this one. Okay, and they may have similar functions or distinct functions, sometimes opposite functions also, sometimes maybe non-functional as well. They can be constitutive. Alternative splicing can be constitutive or regulated. Constitutive in the sense it happens continuously and randomly, and the other one is regulated by the presence of other factors, specific factors. So some of the examples we have discussed about troponin, alpha troponin and beta troponin. Either if 3 is included but not 4, then you get alpha troponin. If exon 3 is not included but exon 4 is, then it is beta troponin. We also discussed about capital T antigen and small t antigen, right? And here what happens is, if in the normal one, the exon splicing happens between 1 and 2 perfectly, and it produces a open reading frame from here to here. Start codon and stop codon will be here, and so you will get capital T antigen, which is large compared to the smaller one. Why is the smaller one small? If you see at the level of mature mRNA, you will find that the one that is spliced together is smaller, whereas the one that is improperly spliced, basically here splicing is happening between these two, or the joining, right? Exon 2 is spliced into an internal site, which is a 5' something like pseudo 5' splice site. So this part of the intron is retained, because of which the mRNA is larger in size compared to that of the capital T mRNA. But the problem point why capital T antigen is larger than the smaller one, because the reading frame is larger. In the small t antigen one, the reading frame is small because there is a stop codon that is in frame with exon 1. Whereas in the capital T one, you have a start codon to stop codon. Although the mRNA is small, the reading frame is larger, which is what gives rise to a protein. And there are several other things that I might not discuss, but you should understand that some will have mutually exclusive splicing. Okay, and that I will just describe one of the examples. Maybe I'll take here. That is steric hindrance. When you say steric hindrance, it's like if there is a small space, and this space is for, say, this is a particular sequence. The sequence stretches from here to here. If a protein is already binding here, another protein which might interact with the same site will not have enough space because it's already occupied by, say, protein A, and protein B cannot fit in there. On the contrary, you can also think of it like this: if protein B is already bound here, then A cannot bind here, isn't it? That is something called as steric hindrance. And also it is also mutually exclusive splicing. That means if you see here, splicing can occur between, in A, I'm trying to here, exon 1 and exon 2 splicing can occur, and splicing can also occur between 2 and 4. We will see what is the consequence of it. But it is also possible that 2 is excluded and exon 1 is splicing with exon 3 and exon 3 with exon 4. So there are two different types of transcripts that can come, like what we have discussed in this case. Sorry, in this case, 2 is included but not 3, right? Here, either 2 or 3 is included. Those are something like alternative exons. Here, it's only mutually exclusive, means either 2 will be included or 3 will be included, but not both. Either exon 2 is included in the top case like here, and exon 3 is not included. Whereas if splicing happens based on the dotted line shown in the bottom, 2 is excluded and 3 is included. And how is that possible? How can what determines whether 2 is what determines if only either 2 or 3 but not both are included? One of these is steric hindrance that I explained here. You should look at this place, right? There is a 5' splice site and there is a 3' splice site. And U1 recognizes 5' splice site, and the 3' splice site is recognized by U2AF, RS1, and so on. The distance between these two is not so much. So if U1 is occupying this place, then the one that recognizes the 3' splice site cannot bind here. So, right? There is not enough space for it to bind, so it cannot bind. Or you can think of it also as a branch point. So I will try to erase all and discuss once again, so it will be easier. So now think of it, if this place is already occupied, there is no place for the branch point or 3' splice site recognizing ones. So that cannot bind here. So now what will happen? Splicing will always occur between the proteins that are recognizing the 5' splice site and the proteins that recognize the branch point and 3' splice site. So now these two are there bound, say U1 and the branch point, so on, and then splicing can occur. Here you have the U1, but there is, because of its binding, the other proteins that are supposed to recognize the 3' splice site are not binding. Where is the next 3' splice site? It is here. And then splicing will happen between these two. So that is how exon 3 will be excluded. On the contrary, if you have the 3' splice site binding proteins or the branch point also included, now the splicing can occur between this one and this, like that. It is more complex than what we know. It is a model basically. If this is bound, because of which you will have steric hindrance, and then there will be exclusion of 2 here, and then you'll have 3 included. Okay. Again, the question can come, should be there. The question is something like, yeah, there is no problem in binding here, isn't it? But there will be many other factors that are involved that we can explore it if we require more. But for our understanding, we should try to know what is mutually exclusive splicing. Like in this case, you can see that you have a minor, that is AU-AC splice sites, and this AU can form something like this. I mean splicing, or this is 1, 2, 3, and 4. And so it might skip 2 in this case and then splice with 3, right? In the top phase, if you see, it has skipped 3 and 2 is included. So that is That is how mutually exclusive splicing can happen. There is another one which we will discuss in the later classes: nonsense mediated decay. So here it is about which one is included. Either exon 3 or exon 4 is included. Okay. When there is if so the red line ones is exon 1, 2 is skipped, and 3 is included, and 4 is included, right? So 1, 1, 3, and 4. And there is also other possibility that you can have 1 and 2, 1, 2, and 3 is skipped, and 4 is included. I will come to a discussion later about what would happen if there is what is meant by nonsense mediated decay. For this point, you have to remember something like this. Say, this is an open reading frame, this is the start codon, and say, for example, because of something, you will also see there is a mutation. This is called a nonsense mutation, which means that a sense codon has changed to a nonsense codon, that is a stop codon. That means ribosome will translate this and will dissociate. Termination of translation occurs here. Then this part of the mRNA is not occupied by ribosomes. And in that case, what happens is exonucleases take up and will cleave it up, and the mRNA is degraded. That is referred to as nonsense mediated decay. We will see those in greater detail at a in a later class. So the introns that are present in genes, some of them are very complex. There is what we are discussing it at a superficial level. We are trying to learn only the principles and a few examples. There are many other examples. There are some more examples that I have skipped because we will need a lot more background knowledge before we understand that. So there is also one more thing. If you remember, like we have discussed about VDJ recombination, there we have several V exons, several D exons, and so on. Meaning that you have exon 4. In this case, there is exon 4, exon 6, exon 9, and exon 17. And each of them again have alternatives. That means one alternative of exon 4 will be included, one alternative of exon 6 will be included, and one of 9, and so on, something like that. Okay, you can look at this. There are 48 alternatives of exon 6. That's a huge amount of diversity again, right? So this is about DSCAM gene, that is Down syndrome cell adhesion molecule. I'm not going to ask you about these examples and so on, but it's an important thing to keep your mind open and not be fixed to what is just learned here. That there are so many other possibilities and complex organizations of genes and the processing of RNA. Be open to those. In the future, if you come across and hear that, say for example, transplicing, I never heard of it before, that two RNA molecules can be spliced together, right? So it's interesting. It is interesting and sometimes astonishing, and that is where we have to keep our mind open so that we can try to understand what is happening or how complex it can get. If you close up, oh, this is the only thing that I learned, this is what I learned is absolute, then we don't have, our minds are closed for future possibilities. So I can tell you an example of where I made a mistake of what I'm saying right now is there was a meeting, and then within the meeting, the student was presenting for a PhD something. And they're presenting, and in mitochondria, those mitochondria which have circular chromosome, circular DNA, some process happens, something like that they was describing. I asked, how is it possible? What else is possible? Bacteria, mitochondria must have come from bacteria. Bacteria have circular chromosomes, so mitochondria will also have circular chromosomes. Until the candidate explained to me that there are there are some mitochondria with linear DNA as well. And then I had to look up the literature, and I found that there are linear DNA. In some of the mitochondria, they can have linear DNA. So what I'm trying to imply is that we just learned about exons and one exon and splicing and so on. But at the same time, now we are seeing alternative forms of each exon, and only one alternative form is included. That is adding a lot of complexity to what we are learning now. So keep your minds open. Just be ready to be uh, surprised. So there are in regulated alternative splicing. We haven't touched about it. We are not going to discuss so much either. Maybe in the review classes we will take up. We'll see if we should. So the regulatory proteins are activators and enhancers, which will either suppress splicing it in a certain region or may activate splicing within a region. I want to spend a little bit of time because it will be directly useful to you if you are taking stem cell biology and so on. And also, it's important to know that. If you have ever wondered, um, if you've been, we have certain cells. We have so many of these cells, right? We come up from zygote, then we form a blastula, and then gastrula, and then we form the adult. And within that time, in an adult, we have so many different types of tissues. Some of them are like the neurons extended. Some of the pigment cells are something like ghostly. You can think of it like that. I hope you understand that there are different types of cells. And what about their genes? Is it same? Is the genome, the number of genes they have, is it same or different? They are the same. The genome is content is mostly the same thing, but the cells look so different. How can we compare? If you have the same genes, why is it not having the same shape? Cells should have same functions and shape, but that's not how it is happening. That is where um, that is when we discuss it happens because of epigenetic regulation. I think I have given you a brief explanation, brief definition of what is epigenetics. Epigenetics is change of, or it is because although these cells have the same genomic material, they have different functions because of regulation. And that regulation is, although they have the same genes, they are regulated. The sequence level change is not happening. If there is a sequence change, level change, then you call it mutations. But instead, there can be DNA methylation. We also discussed about histone acetylation, methylation, and they can suppress some regions from expressing some. In some cases, they might allow the expression of certain regions. Based on that, in different cell types, you have a different set of genes expressed, and based on that, the form and function of the cell um, it is the consequence of the regulation. Here is one of the proteins that regulates that. So if you have cell, this is zygote, okay, zygote, and then they will form the blastula. In the blastula, you call this there are two different types of cells. So okay, I want to take a little bit, I will go a bit back so that I'll be able to explain properly. Okay, here we have a zygote. This is a single celled diploid cell, and that zygote will divide continuously and make a structure called as morula. Morula, the point is that all the cells are identical, okay? And when you when they form blastula, then they form two different types of cells. One is the it's like a football like cell, a football like structure, where you have the outer ring of cells, and then you have a small inner cells. So first time within during this early development of the embryo, we are seeing the cells in two different forms. Genome is same, but they are forming two morphologically distinct cells. This is trophoectoderm cells and or trophoblast cells, and the inner one is called as inner cell mass. They are, although the genome is same, they are two functionally and morphologically different cells. That process of formation of two different types of cells, despite bearing the same genome, same set of genes, is called as differentiation. Okay, all these cells you see, they are all highly differentiated. What you call from the zygote till morula, you call it as undifferentiated. The ICM, all our cells in our body come from ICM, that is the inner cells mass. We have about 210 different cell types, and stem cell, these ICM is the source for embryonic stem cells as well. Okay, if you take them out and culture them, you call them embryonic stem cells, and they are pluripotent. Pluripotent means that means they're able to give rise to complete set of complete or any type of cells within our body. They can give rise to cells that come from ectoderm, mesoderm, endoderm, and also they can give rise to germ cells. So basically, pluripotent means they can give rise to any type of cells. And that means assume that we start where somewhere here, this is should be the ICM, and ICM will develop into the primordial germ layers, that is ectoderm, mesoderm, and endoderm, and they will further divide or differentiate to form different types of cells. So pluripotency is the ability to give rise to any type of cells. That is pluripotency. What I'm trying to describe is this: pluripotency is the ability to give rise to all types of cells. Differentiation means it is forming a functional cell. Functional cell, right? It is differentiating into a particular structure and morphology so that it can perform a function. Something like the stomach cell or thyroid and lung cells that arise from endoderm, they have a function. They have to increase their cell surfaces so that they can absorb, and in lungs they could be able to gaseous exchange and so on. So there is a protein called FOXP1, which is important for switching between whether it should remain as stem cells or pluripotent, or it should differentiate. The pluripotent cell or the ICM by itself is useless. It does not perform any function. If only this cell differentiates into a particular cell type, then it can perform a function. And this state is called as differentiated state. And the stem cell or the ICM is in a pluripotent state. It has the potential to give rise to other cells, but by itself cannot perform any function. Or you can think of it as the function of a pluripotent stem cell is to give rise to differentiated cells. Okay. So when does a cell decide, or how does a cell decide to divide and differentiate into a functional cell? That is dependent on FOXP1. It has several exons. Out of you have 17, 18A, 18B. These two are kind of belong to the exon 18, and you have 19 and so on. We are seeing a part of the gene. In stem cells, through alternative splicing, we have 17, and 18B is included, but not 18A and 19. Then you have a function, a protein or mRNA is like FOXP1-ES, embryonic stem cells probably. And the if this protein, if within a cell, if there is a formation of a mature mRNA consisting of 17, 18A, and 19, then you will have differentiation genes will be expressed, pluripotent stem cells will be suppressed. That is when the pluripotent stem cell will go back to. I will just take a minute to. Oh, sorry. I'll just clear it up now. I hope you have enough introduction to what it is. Now I'm telling you this. You have embryonic stem cell or ICM, which is pluripotent. And it has to form some functional cell. If 18, if Fox P1ES, that is, which includes the 18B, is produced, then it will shift to being, that is, it will allow the production of pluripotency genes and suppress the differentiation genes. So the cell will remain pluripotent. Okay. But if there is an inclusion of, in during splicing, if there is inclusion of 18A, then the pluripotency genes are suppressed. This is how you show suppression. This is how you show expression or activation. And the arrow in 18A, if it is included, that forms the FOXP1, then you will have the formation of or the expression of differentiation genes. And now the pluripotent stem cell will give rise to a differentiated cell. Okay. If you are going to read stem cell biology, you will learn a few more things or a lot more things about this. But I think this much of understanding should be involved. For our present course, what is our focus? Our focus is only about the molecular mechanisms of splicing. So we had this a brief discussion, because why do we introns? How did they come about? Why did they come about? First of all, second is question is, when did they come about? Is it introns came? Is it that all the genes had introns first, and then they were lost in some, or is it the other way around? That is two theories: introns early and introns late. Some people believe that introns, first genes had introns, and later first genes had introns, and later in prokaryotes, which will have to feed fast and grow fast, and their nutrition is limited. In that case, they would not want to spend time on synthesizing the pre-mRNA, then splicing it out and all those stuff. Instead, if they got rid of, if introns were lost, they will be able to grow faster. And that is how it is thought that the earliest genes had the introns, and in prokaryotes they lost introns. Introns late is thought to be that the first genes were introns less, and they did not have any introns, but eventually during the course of time, they think the genes have acquired introns. Okay. And how did they acquire? Why did they acquire is again a question. Some people believe that the acquisition of introns will allow gene divergence, increased gene divergence. And before going that, how did they propagate? I mean, we read that about in humans at least, about every gene has about 7 introns on an average. That means that's a large number of things. And how did they move? How did different genes acquire introns? They think that it is through transposition. Somehow it has occurred. These are all questions, and we don't have clear-cut answers. People can only speculate or build models and try to understand what is happening. Okay, but nobody can be absolutely sure about what is there. There are no evidences. There are, at least in our technology frame or framework of understanding, we cannot decide them clearly. But I have an opinion. This is my opinion. I think I believe that it is introns late. I think if we think the evolution has happened in this direction, that we all have evolved from prokaryotes to eukaryotes, the earliest of the prokaryotes will have a lot of trouble getting resources. That means there is always a constraint on how efficiently you can use the resources. In that case, having something like splicing is a very expensive to have, and not something like that will not allow the evolution or to be even competent along with those that do not have. So I believe that introns have come later in the course of evolution. And that is because through transposition or other mechanisms, eukaryotes, in comparison to prokaryotes, are much more versatile or fit in their ability to get nutrients, because they can, if you take protists, right, the unicellular eukaryotes, they just engulf, they actively move towards a prey, engulf and eat a lot of huge amount of food. They have mobility, so do bacteria. But I think if you, there is a nice YouTube channel, I will try to share it with you, and if you want to be surprised and astonished, you can watch that. I will share the link on the WhatsApp group. I think it's called something about microcosms or microcosmos, I think. Anyway, and is there a use of these introns? I don't really know, but I will have an argument something like this. If there is a gene, and then there is another gene, and it has to recombine. So one gene is with introns and the other one is without introns. Okay, this one has the introns here, and this one does not have. Say, I don't know if this is true or false. I'm still thinking about it, but I wouldn't mind sharing, even if it is faulty, what I think about it. You don't have to believe or disbelieve it. So if there is a break here, right? If there is a break here, there is actually, and there is a break and repair, say for example, there is really no loss of it, right? If there is a break here in the exons, within the exon, if there is a double strand break and repair, some bases might be added or removed, because of which the reading frame could be faulty. You take a gene without introns, anywhere within the reading frame you have a cut, there is going to be a problem, because you're likely to end up having a mutation. So having these kinds of buffer sequences, I just make a phrase like the buffer sequences, where even though mutation occurs, there is no big impact on the reading frame at the end. When splicing has happened, 1 2 3 exons are here, there is no issues, right? So I think while I'll also believe the gene divergence can also is also of using the same logic. If recombination has to happen between two genes like this, okay, between this one and this gene, and if there were any, if recombination happened, say homologous recombination, but there are differences, and then genes might get troubled. They might if they get into any mutations, then they might be, it might be risky to have recombination happen there. Whereas these regions, they are much more they do not have much any impact on the final mRNA, right? So at the gene level, recombination can happen in these locations and it doesn't change anything. That is where, when we come to exon shuffling, that is why I was taking a little bit of time. Exon shuffling is that. So this is at the level of say the RNA, And what people think is, or they have noticed it, is one exon encodes one domain. That is what I have also described some other place, some other time. So typically, one exon is encoding one domain, and when it is upon splicing and then translation, we have a functional protein like this, a protein for a domain for DNA binding, and a protein for domain for dimerization. So you would have the dimerization domains like D2 will be like this, and here are the DNA binding domains, right? And so each one is D1 is coded by exon 1 and D2 is coded by exon 2. So if not in all, in many locations you would find that one exon is coding for one domain. Now if you think of it like that, you can take a gene of LDL receptor. It is made up of different parts of the genes, like recombination has happened and now you have a combination of genes. If you see the LDL receptor, low density, here it is, low density lipoprotein receptor. It is made up of some part of complement gene and some part of epidermal growth factor receptor and some actual functions, right? One gene is made up of different parts of other genes, and that is only possible if recombination happened within the intronic regions, right? Here is the border, and intronic regions you now have combinations of exons. Here is what is also called exon shuffling. I wanted to say that, right? What is exon shuffling? Like this, you have, these are exons, exon 1 and exon 2. The same, similar, somewhere you have another gene which may have exon A and exon B. You can now shuffle these exons. Okay, I will say this one codes for DNA binding, and this one codes for dimerization, and this one codes for binding to actin binding. I'm just making up. The domain A is for actin binding domain. Domain B is I'll just put up nuclease. That means DNA endonuclease. I'll just put like that. Now here is one gene, and this is another gene. Can we shuffle these exons and see what happens? If recombination occurred between them, you might end up like this. Exon 1 can be recombined with exon B. What would you get then? You will get a protein with two domains. One of it is DNA binding domain, and the other one is endonuclease domain. Do we have a new protein now? Yes, this is a new protein, a hybrid. In case if recombination happened between these two genes and something like this has formed, exon 1 and exon A, then it has a new protein again, which will have DNA binding domain and actin binding domain, right? This is the term exon shuffling, is because introns are present, you can have liberty of recombination happening, and as a result you have so many different types of genes, genes forming, because of which those types of proteins, versatile proteins, could be formed, or diverse proteins could be formed. I hope you understood what is exon shuffling. The same thing is what is depicted here. I hope you understood that. So each one is one bromodomain, chromodomain, zinc finger, SWI2, histone methyl transferases, and so on. You can have different combinations and you will have different proteins formed. So the important one in this part is I want you to learn about RNA editing. I'll straight away get into that. What is, I will just say, what is RNA editing? RNA is transcribed, you form a pre-RNA, splicing may have occurred, and now you have mature RNA, right? Mature mRNA, which can further be acted upon, and then can be base changes, sequence changes. And this happen by, so one of it, two methods is what we are going to discuss. Of course, it is RNA editing is change in sequence of RNA. That is, you should not count splicing in that. That is, apart from splicing, there is still sequence changes, and that is all those mechanisms come under RNA editing. One of it is site-specific deamination, and the other one is guide RNA directed modifications. We will see those. Here is a for apolipoprotein, which is also involved in lipid transport within our body. It is produced, it has so many exons, and in one of the exons, in exon 26, there is a codon at that is codon 2153 codon number. So upon splicing, the introns are removed, exons are joined, and this is CAA, which is the codon 2153. And there are two possibilities of what can happen. Without editing, it will be translated and a large protein is produced. It has about 4,563 amino acids, and CAA is coding for glutamine. Okay, upon translation, you'll get such a large protein. But if it is edited, and the editing is happening is cytidine deaminase. Cytosine, you have, we think we will just see if we can make it up. And we have amino group. I'm not drawing the, and then something like this. Okay, 1, 2, 3, 4, 5, and 6. I hope I'm right. Yes. So cytidine deaminase, the amino group is removed and replaced with O. That will result in uracil. Cytidine C becomes uracil upon deamination. So if this C in this CAA, that codon, is acted upon by an enzyme called cytidine deaminase, the uracil, the cytosine is modified into uracil. It doesn't happen anywhere randomly. It only happens at certain places, and that is called as that is what is site-specific deamination. So because of which now it is a stop codon. This is a functional codon CAA, sense codon, which produces glutamine, but now it has changed to a UAA, which is a stop codon, and then it will, the amino acids, one second, TGA, THE, is that right? Yes. Then upon translation, the mRNA will be read from here on only till here, and so it will produce a smaller protein. And you have two different proteins. Either it forms a larger protein like this or a smaller protein, and they have different functions. The larger version of it will transport to the liver, whereas the smaller one will transport to the intestine, the lipids that will be transported from the body to different locations. Okay, I hope you understood RNA editing by deamination. So this is the chemistry, and this is cytidine deaminase. Cytosine replaced with removed, and the keto group forms. And then there is one more that we see is adenine replacing or deamination of adenine. That is called as adenosine deaminase acting on RNA, ADAR. Okay. So it is involved in deamination of adenine. It results in the formation of inosine. Inosine can base pair with any of these. So when we read about translation, you will try to figure out how ribosome reads. And then there is a difference in what happens. That is another chemical modification of the RNA. This is something even more interesting. It is by guide RNA. So what do they do is, here we have seen that one in the last one we have seen CAA, and because of chemical modification it became CUAA. There is no big deal in that, right? Kind of changing just one group of one base you remove and it changes to something else. This is even better because it can insert nucleotides into the RNA. There are also places where you can delete, or and sometimes you can insert. So you see that all of RNA editing, I want you to also appreciate this. This is one mRNA, this is one gene, and it is producing two different types of proteins. One gene is producing two different types of proteins. That is with editing. That is another way of that makes it possible. I'll go to this place where you can explain these things properly. So how insertion happens is it requires something called as a guide RNA. Guide RNA are somewhere around 40 base nucleotides. They have three different sites. One of it is called the anchor region to go and bind to the certain region. Then you have ability to find out which site should be edited. That is the edit site determinant. And there is a poly-U stretch. Those are typically the three things. It can result in addition or deletion. It depends on the specific example. So let's see this, and I will stop with this. So GAG, and this is the mRNA. And what happens is there are, this is the gRNA or guide RNA. It has the anchor region, which has homology to this or complementarity. And then you have editing region, where it is the ability to base pair with the target so that it can bind to that, and that is this site determining where editing should happen. Once it binds here, this U and this U here, they do not have complementary base pairing ones, something like that. Or you can see that when they base pair, it is these two adenines are coming or forming a small hairpin like this. Okay. And that is where an endonuclease makes a cut here. And now two uridines can be added into it in this place. And then ligase joins it. Previously, we had only two uridines, and now you ended up having two additional uridines. You have to remember that anytime we are adding one or two bases or removing multiples of which are not multiples of three, you are changing the reading frame. The reading frame changes in the RNA. If the reading frame changes, what changes also? The protein sequence during translation also changes. So a completely different protein could be formed. Frame shift mutations. When we read about translation, I hope we will start with that. So maybe in tomorrow's class, we will try to have discuss what are frame shift mutations and other things. So try to read about mRNA transport, or maybe I'll see if I have to make anything in the next class. Okay. I'll stop here. Maybe I will repeat this part from here. Yeah, maybe RNA editing onwards I will take up, and RNA transport, and then we'll have a discussion about or introduction to translation in the next class. Any questions?